There's a lot in the news lately about something called pathogenic priming, which is a very scary notion about what might happen to people if they've gotten a so-called COVID vaccine. The idea of pathogenic priming that comes from mostly from a ferret study, we will take a deep dive into what actually happened to the ferrets. And it's not only a good lesson in understanding science and pathogenic priming, but in general, just how this research is done and how ridiculous it all is.
Welcome, everybody. Thank you for your patience. Let's get started. All right. Welcome to this special live presentation with Dr. Andrew Kaufman and Dr. Tom Cowan. Today's presentation is titled COVID-19 Myths, Pathogenic Priming, Genome Sequencing, and the New Genetic Vaccines. Dr. Tom and Dr. Andy will present brand new material today. The webinar presentation will be followed by a live Q&A with both doctors. Welcome, everyone. Let's get started. Good morning, doctors. How are you?
I'm good. I'm good.
Andy, check your microphone.
I have my mic turned off because Tom is going to start us off today, right?
Yeah, I thought you were going to say something a little bit, but maybe you already said it. So,
well, what I can certainly say something if you like. So let me just tell everyone. That's strange. The outline of what we're going to do. So we're going to kind of take turns and do a little tag team. So Tom is going to start us off talking a little bit about the history of the science of virology and some of the key experiments. And then I'll expand on that and talk about a new experiment done by Dr. Lanka and his team. And then we will shift gears back to Tom, and he's going to then introduce the pathogenic priming issue and talk about the ferret study. And then I will wrap up talking about the genome sequencing and the new vaccine paradigm. So to make sure everyone leaves at the end thoroughly confused. And then we will take some questions after that. And I know we have a lot of questions already. So we'll be ready to go with that. So with that, Tom, why don't you
yeah. Okay. Thanks, Andy. Just to say, I also apologize for us being late. I'm not somebody who's rarely late. I usually tell people if I'm late somewhere, that's probably something has happened, because I don't really like to be late, and I usually start on time, but these are unusual events. So with that, again, thank you. It's really an honor to speak to you all. And the first thing I'm going to say is not new, but it really is necessary in order for Andy to put the Stefan Lenka study in its proper perspective, because it really is a revolutionary study. And I know that some people have probably heard what I'm about to say, but I think it still merits going over, hopefully as briefly as I can, just so we can see how important this study actually is. So the question, and I would submit after a year plus of dealing with this, ever since my original video in March or April of 2020, what it's come down to is a very simple question, which I sort of ask everybody, including any medical doctor, any virologist, any layperson, anybody I meet who's interested in this, that all you need to know is the answer to the question, how does a virologist identify a new virus and then show this virus causes disease? That should be on the same kind of level as, you know, if you want to hire a baker and you ask them, how do you bake bread? They should be able to say, I start with 100 grams of flour and I add water and I knead it or add salt or whatever it is. I'm talking about the exact steps. And it should be that every medical professional, every medical doctor, and really every human being, because we're all affected by events which are all based on the existence of a virus and the theory that this virus causes disease. So everybody out there needs to know how they came up with this. And then basically what I'm going to tell you is how it should happen and then how it does happen. So let's just get right into it. So if you start by asking, or if I've started by asking, which I have, literally hundreds of lay people, how do you identify the existence of a new virus and show it causes disease? They usually say things like, well, a lot of people get sick in this place in Wuhan or New York City or Northern Italy, and so that proves it's a virus. Or they say, well, a lot of people got sick in this place and then it spread to another place, and that proves it's a virus. Or there was people, nobody was sick in the prison, and then somebody tested positive with a PCR test, and then a new bunch of prisoners came, and then they all got sick, and that proves it's a virus. Or my Aunt Bessie went to church, and then she was fine, and somebody had a positive test in church, and then Aunt Bessie got sick, and then that proves it's a virus. The thing I want to make sure everybody knows at this point is if we're going to be scientific, if we're going to be scientists, these are all epidemiological observations, and the role of epidemiology in science and medicine is not to prove the existence of anything or prove causation, but suggest hypotheses that can be tested, and period. And there is no discussion about this. There is no virologist or competent medical doctor who thinks that any of these observations prove the existence of a virus or prove that it causes disease. So please don't make the mistake and use that as an explanation. Or the idea of, well, what other reason are people getting sick? That's simply not how science is done. And as I pointed out many times, if you think that because a lot of people get sick in the same place, that proves it's a virus, you must think that Hiroshima was a virus. And if you think that if it spreads from one place to another, that proves it's a virus, then you must think that Chernobyl was a virus. And as I've also pointed out, for hundreds of years, people observed that sailors got sick on ship, their teeth fell out, and then they went into heart failure and died, one after another, and then sometimes the next ship, and they thought it must be something spread between one sailor and the next, which is a reasonable hypothesis, but it turned out they had scurvy and somebody ate a lemon or a lime and the whole thing went away. We've had many examples, beriberi, pellagra, etc., where people thought for hundreds of years there must be something contagious, and it turned out to be a whole different explanation. So let's none of us ever again make that mistake. So now we move to the next level. And this is a little more complicated, but it's also square. I'm going to tell you the number of people, the types of people and institutions who agree with the following statement. The statement is, in the history of published science, medicine, and virology, there is not one example that you can take a sick person or an animal and show that that sick person or animal has the same morphology, meaning shape and size, of any pathogenic virus in any bodily fluid, whether it's from the lungs or the snot or the blood or the cerebrospinal fluid. There simply is no case that that's how it's done. In other words, if you take somebody with polio or measles, you cannot look at any of their fluids and find thousands, hundreds, millions of identical particles, same size, same shape, same genome. That has never been done. Now, that process is what we would call isolation. In other words, you take a sample directly from a patient, and then you do the steps of macerating, filter it, ultracentrifuge. You come out with a band. You see thousands of identical particles. You do an experiment to see or an analysis to see what they're made of. You find the genome. And then you could take those particles that are purified, isolated virus, and then expose an animal or maybe a person to that virus in the usual way, like spraying up their nose or something like that. And you would expect them to get sick. And that is the proper way to isolate, purify, identify, characterize a virus. So you would find a new virus, and then you would show the virus causes disease in an animal. And again, I can tell you that process has never been done with any pathogenic viruses. Now, let me give you a short list of the people who agree with that. Number one, 58 different government health agencies, when asked under Freedom of Information requests, say they have no paper describing the isolation and purification and characterization of any SARS-CoV-2 virus from any human being with so-called COVID-19. We also have the same Freedom of Information requests from many governments, not 58. The same thing, no HIV has ever been found from any person with AIDS. No measles virus has been found directly in any fluid with anybody sick with measles, and on and on and on, Zika, Ebola, etc. All 58 governments agree. 15 different institutions like the CDC and the Robert Koch Institute and the Pasteur Institute all agree with that. They say that is not how we find viruses. We cannot look in any fluid of any sick people and find identical particles that we would properly call viruses. I would also point out that this is not because that viruses, meaning particles that have a protein coat with a genetic core of that size, cannot be found, because that's simply not true. For decades, we've been able to find so-called bacteriophages, same size, same morphology. They have protein coat, genetic interior. We can isolate them and purify them directly from bacterial cultures. Stefan found a so-called giant virus in a sea algae, exactly that way. And as Andy has pointed out numerous times, exosomes can be found, same size, same morphology, same type of genetic material in exactly this way, taken directly from a sick person, macerate, filter, ultracentrifuge, show the morphology, characterize the genome, and it's simply not able to be done with any pathogenic virus. The other people who agree with that are virologists, who say there isn't enough virus from a sick person to actually find it in this way. And Andy and I happened to be on a call with a gentleman who was there sort of to debunk us, who was a senior virologist, at least that's how he was introduced, from this Chinese CDC in Wuhan, and then a Yale virologist. And we asked, can you find this SARS-CoV-2 virus from any fluid from any person with COVID? And he said, no, there's not enough to see. He said, you have to concentrate it. So Andy asked him, what if you put 10 people with COVID? Would there then be enough to see? And he said, no. And Andy asked him, what about 100 people? And he said, no, you have to concentrate it. And then we asked him, what about 10,000 people? And he said, no. And then he wouldn't answer for 1 million people. But the point is, nobody thinks you can find a virus directly from the fluid, any fluid of any sick person. Every virologist would say that is not possible. That is not how we find viruses. So we should be all very clear about that and not make that mistake. So the next question is, well, how come there's, I would say, thousands of papers, of which now I would say, unfortunately, I've probably read 20 to 30 of them, that say isolation and purification of such and such a virus, including SARS-CoV-2. Why do they say that? Or what did they do that would make them say that? And again, we've been over this many times, but they do what's called a viral culture. This was started by a guy named John Enders in 1954. He wrote a paper. He was given the Nobel Prize, I think, in medicine somewhere along that time. And he was the first one to do this, because at that point, the virologists realized they couldn't find any pathogenic virus, any identical particle from any sick person. And believe me, I've looked, and it doesn't exist. So what he did was he was studying measles. He took the secretions, the snot, from somebody with measles, a child, and then he washed it a little bit, and then he filtered it. Now, that is not purified, isolated virus. It has everything that's soluble from a person's mucus, which includes proteins and DNA and RNA and any kind of virus that may be in there, exosomes, and probably a lot of other things. And so he took that, he mixed it with milk, he mixed it with fetal bovine serum, horse serum, and something else like that. And then he inoculated that on monkey kidney cells called vero cells. And then the vero cells broke down what's called a cytopathic effect. And that proved to him that he had isolated the virus. And this is the point I want everybody to understand by the end of this session. The way a virologist proves the existence of a virus, what they call isolation of the virus, is the fact that unpurified secretions or blood or urine, it could be any fluid from a sick person, mixed with other genetic material, and then the nutrients are withdrawn, and then antibiotics are added to this to kill the bacteria, which shouldn't be there because you already filtered it. The fact that it causes breakdown of the culture cells, a so-called cytopathic effect, otherwise known as CPE, is not a way that virologists prove the virus exists. It's the way. Every single genetic sequencing, every single experiment done is done with this as the first step. That's called the isolation of the virus. And every other experiment is done after this viral culture. The viral culture is the point we have to focus on. Now, interestingly, Enders did a sort of control where he did the entire experiment. He took nothing from anybody with measles. He mixed it with horse serum, I think, although he didn't specify exactly. He mixed it with the fetal bovine stuff, put in antibiotics, withdrew the media, and he said the results he got were, quote, indistinguishable from the experiment in which he started with somebody with measles. Now, let me just say that again. He did two experiments. One, he started with measles. One, he didn't. He said he got the identical results, and somehow that was overlooked because that tells you that the cytopathic effect, that which we call the isolation and proof of the existence of the virus, happened not because of anything from the child with measles, but because of the experimental technique. That's what this whole thing rests on, that very fact. And so Stefan appropriately said, okay, let's repeat the experiment, and we're going to start with nothing, just like Enders sort of did, and we're going to see what happened. Because if we get the same cytopathic effect, that actually proves that the cytopathic effect is not because of a virus, which they're not even testing a virus, they're testing soluble material from a sick person. So even then, it's not really a good experiment. But even granting them that, he wanted to see what would happen if he repeated the experiment, nothing from anybody added. And I think with that, I can turn it over to Andy and we can find out what actually happened with this test so we can see where we're at. Thank you very much.
Well, thank you, Tom. That was really excellent, and I'm definitely going to back you up on a few of those points for sure. And I'm going to present some slides here and kind of give reinforce and emphasize some of Tom's points, and then we'll get to the control experiment. All right. So hopefully everyone can see that, and we'll get going. I forgot. I wanted to remind people that in the tabs on the right side of your screen, there is a files about 3/4 of the way down. And if you click on that, you'll see the handouts or the bonus materials that are available for you. If you want to grab them while you're here, you'll still have a chance to get them later, but they are available, which is my slides and Tom's references. Okay, so let's get started. So to back up Tom's point, why is this the most important question? In other words, the question of the existence of the virus, the de novo claim. And if we look at Carl Sagan here, said extraordinary claim requires extraordinary proof. Now, there's an important point here because many people kind of ask me if I have a theory or if my theory is better than, other theories, or I've just, proven something, or all these kind of things. And I want to just make it clear that what we are doing here is we're actually examining someone else's theory, someone else's claim. And the claim is that there are microscopic particles which have never actually been seen. They emerged out of nowhere, spread around the world, and caused the worst disease in 100 years. And we're simply saying, where is the evidence to back up that claim, rather than making any claims of our own? Now, according to this Hitchens razor principle of logic, what can be asserted without evidence can be dismissed without evidence. So this goes for the proposition of the virus. And many times when we are in a discussion, like Tom mentioned when we were presenting to a group of other doctors and scientists, most likely these are the arguments we get back, which are actually not direct arguments themselves, but what's known as logical fallacies. And I'd say that ad hominem is the most common one, certainly that I hear from people, which is trying to undermine your opponent by questioning their credibility or their skills or their training. So for example, if someone would say that, we are not virologists, well, that is true, but that really is irrelevant to the scientific discussion that we're trying to have about the isolation and culture experiments. And then it's important that whenever you look at a scientific paper, you have to have this appropriate context, which is that more than likely the paper you're looking at presents false findings. In other words, their scientific conclusions are not true. And this is with more than half of published research papers, and this comes from the scientific establishment themselves admitting this. So whenever you read a paper, you have to have this in mind. Is this even a valid paper, right? It's got basically a 50-50 chance, or slightly biased in the false range. And if it's a virology paper, then most certainly it's on that false side. So the virus principle is really the root of every single piece of tyranny that we're facing, right? With respect to the tests, the injections, business closures, lockdown, every single thing is a result of the virus, which is why if you can simply understand that it's not even in existence, then all of these things are very, very simple to navigate. Okay. And then here's just kind of a summary of my reasons before we get back into the science. Okay, so let's get back to those experiments. So now this is the way that you would do a proper virus isolation, where you get the virus directly from the body fluids of a sick person, which we can see here in the top left, and we would essentially do purification steps on that body fluid. So first we'd filter it to get all of the big things out. And then we can use a variety of techniques. The time-honored standard technique is this density gradient ultracentrifugation, where you have a test tube that has different concentrations of sugar or sucrose, which have different densities, which is the ratio of mass to volume. And those are layered, and then you put your sample material on top of the test tube and spin it at a really, really high speed. And the different things in your sample will go to the place in the sugar solution where they have the same density, and they'll form a band there, which is represented here by this dotted line. And then you can put a syringe or a pipette right in there and suck that out. And then what you would see is particles like on this slide that all are the same size and shape and appearance, right? In every way you see these particles are identical. Now, if we zoomed out on this microscopic slide, you'd see that there'd be thousands of them just like this, and you wouldn't see anything else other than this small particulate debris in the background. Okay, so you'd see no big cells or any other types of small particles like this. You'd just see these particles that all look identical. And these particular ones are bacteriophages, which have been commonly isolated by this procedure. In fact, dozens of them have been analyzed this way. And then, as Tom said, once they're purified in this state, you can examine what they're made of. You can pull out the genetic material if there's any, and you can sequence that directly. You can do different tests on them to learn different things about them. Now, of course, it's important to always have a proper control experiment to make sure that whatever you're looking at came from where you thought it came from. And so in this case, it might be like taking a healthy person and using their body fluid and putting it through the same exact process. And then you should find essentially a blank screen, right? Because there are no viral particles there. Now, what they actually do, since they've never done that, as Tom pointed out, is they do this cell culture or viral culture experiment, which they call virus isolation, with the special meaning of the word isolation. And they start with the same clinical sample of a sick person's fluid, except they don't purify anything out of it. They may filter it or do a basic centrifuge, but no density gradient, nothing that would purify and get particles that are all the same thing. And then they put it in this tissue culture. Okay. And in the tissue culture, which is usually foreign cells, occasionally it might be human epithelial cells, but most commonly it's kidney cells from a monkey. They use the depleted nutrient diet, the starvation diet, antibiotics generally at higher than standard concentrations, and fetal calf serum, which is really an additional source of nutrition, but they lower the concentration of that during the protocol. And then they get these CPEs, or cytopathic effects, as Tom pointed out. And this is the proof that there's a virus back in this snot here, okay, according to virologists. But as Tom said, they don't do a control experiment where they leave out this part, but do the tissue culture exactly the same to see if they would get the CPEs. Okay, so this is never done until now, except for possibly the Enders experiment. So this is Lanka's control experiment design. So he just had the cell culture. In other words, he didn't put anything in it, any body fluid from any animal or human being at all, but had the same culture conditions, starvation diet, antibiotics at higher than normal concentrations, and fetal bovine serum. And guess what his result was? He also got CPE. So just to remind everyone, this is supposedly the proof that there's a virus in your sample. And this result occurred here with no virus in the experiment whatsoever. So what this is telling us is that the experimental procedure itself is giving us this alleged proof of the virus. Now, here are the actual images from Lanka's experiment, and I'll explain this. There are 4 columns here, as you can see. This is the control of the control. So this whole experiment is a control, right? But he has actually a regular control group in here to compare different ways of growing the cells. So this one has culture medium, which is the full spectrum nutrition, okay, plus 1x, that's 1 times the concentration of antibiotics, and antimycotics, which is antifungals, okay, and specifically it's amphotericin, the same drugs that are used in all of the COVID-19 SARS-CoV-2 experiments that Tom and I have discussed. So this is what it looks like one day after the stress, and the stress is just changing these nutritional conditions. Now you can see this looks the same as the other ones looked at the start, because this is what a super healthy cell culture looks like, okay? And you could see in that group, 5 days later, it still looked super healthy. Now let's go to control group 2. Now this is closer to what the virologists do in their experiment, but not quite. So here they changed to the different type of nutritional medium called DMEM, which is Delbecko's modified Eagle medium, or sometimes it's called minimal essential medium, and plus 10% fetal calf serum. So this is full strength Delbecko's medium with calf serum, okay, but it's still not as complete as the culture medium. That's the Mac Daddy nutrition, and then this is modified nutrition. So it's kind of like going on a diet, and still with 1 times the concentration of antibiotics. And you can see there is some changes to these cell cultures. There's this area where there aren't healthy cells, okay, and you can see that some of these things look bigger. They're not uniform like in the healthy picture, okay? So something already is going wrong. Now, if you look at the next one, and this is actually how they really do the viral culture experiment, that they reduce the DMEM to, I think, half of its concentration, and they reduce the fetal calf serum to only 1%, so from 10% to 1%, and then they increase the antibiotics, and I've seen that it's either 2 times or 3 times the standard concentration. And then you can see here that these cells go from normal to even more abnormal than the other control, and then this shows cytopathic effects. So essentially we have here, there's no source of the virus at all, and you see cytopathic effects. Now, I'll just mention briefly the last group, and this was really done for a different reason, but yeast RNA was added. Otherwise, the nutrition and antibiotics were kept the same. And you can see that it's even slightly more toxic. And the yeast RNA is benign. It's not anything that would be considered to cause a disease at all, but it is sort of a proxy variable for perhaps a biological fluid that would have RNA, but also many other constituents in it. And it's done for some additional experiments that in future months we'll be reporting on as well. So the significance of this is that this experiment right here completely disproves the theory of virology, or the theory that viruses cause disease is now completely disproven, because this experiment shows that none of the evidence ever provided for a virus being present in a specimen from a diseased person or animal is valid. It's all been a result only of the lab experimental procedure and never from the presence of an actual virus, which doesn't exist. So, to wrap up, if no virus exists, as now has been conclusively shown scientifically, then there's no known structure or composition of a virus like the spike protein. And if you say, well, what about all those studies on the spike protein? If you go and look at any one of those studies, you'll see that they did not get a spike protein by taking it off of a virus. What they did is they used a fake genetic sequence to make a recombinant spike protein by putting that sequence into a recombinant organism like a bacteria or a yeast, and then having that microorganism make the spike protein so they can study it. Number two, there's no known genetic sequence. Of course, you can't have a genetic sequence of something that doesn't exist. There are no genetic variants or mutations. There's no way to develop a test to test for something non-existent, and there's no way to demonstrate there's a cause of the disease. So it's basically gives you the answer to every single question related to the pandemic. All right, Tom, I think we're ready to talk about pathogenic priming now.
Right. So it seems like our lot in life is, I'm talking about Andy and I, because you could say, okay, pathogenic priming. So let me just say, what is pathogenic priming? So pathogenic priming is a theory, I guess I would call it that is circulating strongly amongst this sort of anti-vax community. And I hear it a lot, and I hear it as given sometimes as the primary rationale for why it's not a good idea to get this experimental injection, otherwise known as a vaccine. And I hear it debated amongst sort of normal scientists, whether there is such a thing, and they go back and forth. But the thing that really concerned me, and the reason I wanted to look at it, was because, A, I was getting so many questions about it. Is this something to be concerned about? And is this something that we can use as our primary evidence that like our loved ones or friends shouldn't get this experimental injection, because we can tell them they may get pathogenically primed, if that's a word, and so that's the reason they shouldn't do it. Now, so what does pathogenic priming mean? As far as I understand it, it means that if somebody is given a COVID, quote, vaccine, then the point of it is to make them make antibodies against the SARS-CoV-2 virus, although more specifically they mean against the SARS-CoV-2 spike protein, which allegedly comes from the virus, which we now know isn't true. But let's just go with the normal narrative. The normal narrative is you get a vaccine so that you make antibodies, so you make proteins of the spike protein, and then you make antibodies to that spike protein, And then if you encounter that spike protein in a so-called wild virus or a virus living on the outside, you already have antibodies to it, so you will be able to defend yourself against the attack of the wily SARS-CoV-2 virus. Pathogenic priming means for those who have been injected with spike protein sequences, who make spike proteins, who make antibodies, they, lo and behold, will, if they encounter the SARS-CoV-2 virus, they will make an overreaction. They are primed to make an over-immunological reaction, sometimes called a cytokine storm, and they could even die or have serious injury as a result of the encounter with this wild, free-living so-called virus. And that's a reason why nobody should ever get a SARS-CoV-2 or a COVID injection. Now, it's not really my intention to pick fights with people who are telling people to be cautious of doing COVID injections, because I certainly applaud that sentiment. But there's something about this that really doesn't strike me well, and it partly because it also fosters the narrative that there is in fact a virus out there, which is in fact the cause of all this trouble, and which, if we don't be careful and do certain things like do the injection, the virus will become even more dangerous. And I just don't think that any of that is actually accurate. And that was the first reason I started to look into this. What is the evidence that this is actually true? When I looked at the evidence, I also then came up with another reason for going over in some detail one of the main studies that this theory of pathogenic priming is based on, which is the ferret study. And the reason is because this study dramatically, I would say, teaches us about the way science is done. And I think you will see it in a few minutes exactly what I mean. There was a guy who said the problem these days is, especially in virology, it's all technology and no biology. And, you know, my training, my interest is in, you know, helping sick people in actual like clinical situations. A person is sick and I try to do something to help them not be so sick or be well or better than well or something like that. And one would think that these scientific studies are really all about that. But what you find is they're much more about technology than they are about what actually happens to either people or, in this case, animals. And I think when I go through the study, you'll see exactly what I mean. Now, again, here's the theory. Pathogenic priming, which is often given by the people who are questioning vaccines as the number one reason why you or your loved ones or family or friends should not get one of these COVID injections. That's often the number one reason. Pathogenic priming means if you inject somebody with a injection to help them or make them make antibodies to a component, in this case, the spike protein of the SARS-CoV-2 virus or the SARS-CoV-1 virus, that will make them make antibodies. And then if they encounter the real virus later on, they are primed to have an overreaction, which may even kill them or do them great harm. That's what we're talking about. Now, in at least one of the studies I read, one of the main papers that is given as evidence that this theory is correct is the so-called ferret study. And I have it here. This was published in July 29th, 2004. the title of the paper. It's in the Journal of Virology, November 2004, pages 12672 to 12676. The title of the paper is Immunization with Modified Vaccinia Virus Ankara-Based Recombinant Vaccine Against Severe Acute Respiratory Syndrome is Associated with Enhanced Hepatitis in Ferrets. The lead author is somebody named Hannah Weingartel. So besides that they have slightly poor English, and I'm not sure that they should have had somebody edit the title. The first thing that I noticed with this, again, the thing we're talking about is if you are vaccinated or injected with a SARS coronavirus vaccine, that's the first step in this pathogenic priming process. So the first thing that got my attention is it says immunization with modified vaccinia virus. So it turns out the vaccinia virus is the smallpox virus. So to my surprise, which I thought they would be vaccinating the ferrets with a SARS-CoV-1 virus, but no, they were actually using a smallpox virus as the original vaccine. Now, I would admit then they modified it, so this was a recombinant, genetically modified smallpox virus, of which the genome was extracted from the virus. Now, they don't tell you, and I'm not enough of a virology laboratory expert to know actually how do they take out the genome from the middle of a vaccinia smallpox virus. I must admit, I had visions of some guy in a laboratory with a micro tweezers plucking the genome out of the vaccinia virus shells and somehow throwing them in the trash or somewhere. But I know that that's actually not how it happens. Anyway, somehow they extract the genome out of the smallpox, the modified smallpox virus, and then they insert essentially a spike protein sequence from the SARS-CoV-1 virus. So they insert the SARS-CoV-1 spike protein, the spike protein allegedly coming from the SARS-CoV-1 virus, into the center of the vaccinia shelled out virus. This is similar to what the Johnson & Johnson injection is doing. They're using an adenovirus instead of a vaccinia virus, but same idea, extract the DNA, the genetic material, in this case RNA, from the virus, and squirt in with some mechanism, which I'm not sure how they do that, the spike protein sequence that allegedly comes from the SARS-CoV-1 virus. So that is the so-called vaccine. So how did they do this experiment? So the first thing is they got a bunch of ferrets. They said they got 12 ferrets. And they got them from a place called Marshall Farm Pet Supplies in Wolcott, New York. And they had 6 to 10 week old male castrated ferrets that they used. 12 of these ferrets, they were used in the experiment. They don't say any reason why they castrated the ferrets, and we don't know whether that has anything to do with anything that happened to them, but it's interesting to note. And again, when I'm trying to compare this to what actually happens to like my patients and what happens in real life, you don't see that many castrated males walking around. I'm sure it happens. So that's already an interesting difference. And then they said the ferrets were divided into 3 groups. Remember, they started with 12 ferrets. And here I'm going to read you. The ferrets were housed individually, i.e. 1 ferret per cage, and were immunized with parental MVA, that's smallpox. That's ferrets 1, 2, and 3. Our MVA-S, ferrets 7 through 9, or phosphate buffered saline, ferrets 10 through 12. On day 0, with a dose of 10 to the 8th PFUs of the corresponding virus per ferret by intraperitoneal and subcutaneous routes, and a booster immunization was given on day 14 with the same regimen. So let me dissect for a minute. So they say they started with 12 ferrets 1 through 12. Ferrets 1 through 3 were given just the shelled out smallpox vaccine, no spike protein. Ferrets 7 through 9 were given the hollowed out smallpox vaccine with the sequence of the SARS-CoV-1 spike protein inserted in the middle. Ferrets 10 through 12 were given a saline phosphate buffered control. Now, interestingly, unlike most vaccinations, or certainly smallpox, they gave them 2 on day 1 and day 14. So instead of giving the usual 1 vaccine that is characteristic for smallpox, they gave them each of these 3 different, either just the shelled out smallpox with the spike protein, or a saline control on day 1 and day 14. Now, another interesting thing is they say they gave them by intraperitoneal and subcutaneous routes. And they don't say which one, or whether they gave them one or the other. Intraperitoneal means they gave these injections into their abdomen, which is a highly unusual place to give a vaccine, into the peritoneal cavity. And it sounds like they also gave them at the same time a subcutaneous injection of one of these 3 experiments. The final thing that was interesting is there was supposedly 12 ferrets. Ferrets 4 through 6 have somehow vanished. There is no mention of what happened to ferrets 4 through 6. 1 through 3 got the shelled out smallpox. 7 through 9 got smallpox with the spike protein. 10 through 12 got a saline. They gave each one of them into the abdomen, highly unusual. They gave them 2 at day 1 and day 14. And that was how they, quote, vaccinated these ferrets. Now, the next thing is at day 14, 2 weeks after this day 14. So now we're talking day 28. I'll read this again. It's a little bit boring, but briefly, a triplicate of serial dilutions of heat inactivated ferret sera were incubated we don't need to know that. After incubation, the serum SARS-CoV mixture was added to monolayers of Vero V76 cells. They were incubated and they were stained with formaldehyde and carboxymethyl cellulose. Now, again, they say the challenge then is SARS-CoV-1 virus. That's what we're told, that if you encounter the virus. But as we now know, they didn't give these ferrets intranasally, meaning spray up their nose, any virus. They gave them the results of a tissue culture, just as Andy and I explained in the first part of this talk. So there was no virus that they gave. They only gave them the toxic results of the tissue culture with all the antibiotics and the fetal calf serum, etc. That was sprayed up their nose. So this was, they say, 2 weeks after the booster immunization. Ferrets were challenged with 10 to the 6 PFUs of SARS-CoV TOR isolate by the intranasal route in our biosafety level 4 facility. That's just what I said. In other words, at day 14 after the second dose, so now we're 28 days into the experiment, they took all these 9 ferrets and sprayed the results of a toxic cell culture up their nose, and that supposedly reproduces the exposure to the wild virus that is so concerning in this process of pathogenic priming. Now, at this point, then, they observe the ferrets for another 29 days, right? So after the nose thing, they keep the ferrets in their cage, isolated, which probably isn't so good for ferrets, and they observe them for 29 days. And the whole theory is based on, if you do this vaccination, this immunization, this injection with the spike protein, and then you expose them to the virus, horrible things will happen to them. So now I would like to ask everybody to think for a minute. On day 29, after the nose intranasal injection, what do they think happened to the ferrets? And I'm going to pause for about 2 minutes and see if anybody can put in the chat, here's what I think happened to these ferrets. This is exactly now what we're looking at, called pathogenic priming, vaccinated with spike protein, exposed to the quote virus, wait 29 days, which is a long time for ferrets. What happened to the ferrets? I don't know if anybody can read some of the answers. I've asked this to about 20 people.
I can help you with that.
Yeah.
Tom, if I may, just for a second, something occurred to me when you were describing this, that they were in a bio level 4 safety, right? That means they had to put on all kinds of special safety equipment, right? And if the stuff is so dangerous that you could just breathe a drop of it, you need all that equipment. Why do they have to put it injected inside the abdomen of the, why can't they just put it on the table in the same room or something?
Let the ferrets run around in the cages. All right. Somebody want to read what their guess as to what happened to the ferrets?
The guess is that they all died, and it seems to be everyone is guessing that they died.
Yes. So I've asked, let me say, 30 to 50 people, and I would say 99% of them said either they all died or they all got really sick. Now, there was one person I asked who gave me a different answer. His name happens to be Andy Kaufman. And he said, well, nothing happened to the ferrets. Now, here's the question. Who's right? Because after all, the whole premise is this is a horrible thing. You pathogenically prime them. Of course, they're all going to die. So here's the quote from the paper. Although no clinical signs, for example, elevated temperature and altered behavior, including feeding, were observed up to 29 days post-challenge. What happened to the ferrets? Nothing. So where did this come from? Now, of course, they have some answer here, which is then they did blood tests to see if there's anything wrong with these ferrets. And here's what they said. Surprisingly, quoting, surprisingly, ferrets vaccinated with RMVAS, that's the spike protein one, demonstrated a significantly higher level of ALT after challenge with SARS-CoV than the control ferrets. The elevated level of ALT was evidenced by 4 to 6 days and lasted until 22 days. All the other parameters tested in normal or slightly higher than normal physiological range compared to the reference value. In other words, okay, I looked at the ferrets, I gave them 2 injections right in their abdomen of this toxic spike protein sequence. I squirted a toxic cell culture up their nose. I looked at the ferrets for 29 days. What happened to them? Nothing. So I got to go further. I did some blood tests. A few of them had elevated liver enzyme, and otherwise all their laboratory parameters were normal. And so then they killed the ferrets, and all of them had signs of antibodies in their liver and what they call hepatitis. And the worst of them was one of the ferrets who was injected with the spike protein. And that is the entirety of the evidence for pathogenic priming in this one study. There were other studies, but it's basically the same kind of stuff. Now, the reality is, if you're doing clinical medicine, and I'm not saying by any means that I think it was a good idea to inject ferrets with synthetic spike proteins in their abdomen and subcutaneously, and then castrate them and put them in solitary confinement and give them GMO food and then squirt toxic cell cultures up their nose. I'm not saying that's a good thing. And I don't know where the PETA people are when you need them here. But the reality is, clinically, nothing happened to them. Now, why would they get hepatitis? Well, and especially to have increased antibodies in their liver on autopsy. I think we all know that you make antibodies to neutralize proteins that shouldn't be in your bloodstream. And in this case, because they injected them right into their peritoneum, they probably ended up in the ferrets' livers. And so the ferrets, in order to combat this, made antibodies to neutralize these foreign and potentially toxic proteins. It likely was temporary, just so they could neutralize them, excrete them, and get on with their ferret life. But unfortunately, they were sacrificed, so we'll never find that out. And in other words, the evidence that this is a real problem from this study is only based on very suspect information, which clinically was basically nothing. And it turns out there is a perfectly good explanation for why you would see inflammation and antibodies in the liver, which is that you injected them with toxic proteins right into their abdomen, and that's exactly what you would expect. It's likely it was temporary, and this is no evidence for any kind of pathogenic priming at all. The other study, like I said, was similar, and I just think, besides the fact that we know there's no virus, so there can't be pathogenic priming, that we don't need these kind of explanations. They just ingrain in people's minds that there is a virus, they should be scared, and God forbid you get a vaccine for COVID, and then you're in real trouble. Not that that's a good thing by any means. So I, again, appreciate anybody warning people not to do that. But I just don't see the science of this, and I think we should stick to the science. That is the way out of this mess. Thank you.
Amen, Tom. Couldn't have said it better myself. And so speaking of the science, I'm going to pick up here. And, it's really unbelievable to see that people can interpret this and think that it means something. But, the bottom line is the genetic vaccine is toxic, and it's causing lots of health problems. And we don't need to use mechanisms based on the virus or antibodies to understand that. We can just see what the outcome is. Okay, so we're going to shift gears again, and I know there's going to be a variety of questions at the end, and I'm going to talk about the in silico genome. So just to give a little bit of background, because I know genetics is extremely confusing, and even for the scientists, it's one of the most difficult topics. So I want to just keep this as simple as I possibly can, and hopefully you'll understand. what I'm talking about. So DNA is the genetic materials in our bodies. And it's said that the specific sequence of the DNA is what makes us uniquely human or unique individuals. And by the way, this is the mainstream theory of genetics, not necessarily my theory of genetics. But this code is made up of 4 bases, which are the building blocks of the DNA molecule. And they are represented by 4 letters. And you put these letters in a string, and that will represent the code of the DNA. And what is DNA, or what does it do? Well, this is called the central dogma of biology. Interesting choice of words. Germ theory is a theory, and this is a dogma. But what it states is that DNA is transcribed into RNA, which is a single strand. DNA is 2 strands. And then that RNA is directly used to code for and build a protein. Now, there are many reasons to think that this is not actually the case, but let's just go with this for basic understandings. Now, the way that the RNA makes a protein, and I understand that people are not seeing my cursor, so we're going to look at the image on the bottom of the slide, which shows a ribosome, and you see that there are 3 letter blocks. There's one CUC, CAC, and then UUU. So those 3 letter blocks, those are called transfer RNAs, and they have a 3-letter RNA code. And you see on the bottom, they have another 3 letters, and those are names of amino acids, which are the building blocks of proteins. So this is how the code of the DNA turns into the RNA, which turns into proteins. Now, let's talk about what a genome is. Now, a genome is the entire genetic code of an organism. And in humans, as we know, that these are actually on chromosomes, which are strands that are very, very long. And we have 23 pairs of these to make up our entire genome. So we have, the Human Genome Project, which I mentioned here, which has been undertaken already, and it took quite a while to do. But what they did was they started with an actual person, and just from human cells, they extracted the genetic material. As you can see, these are what a colorized version of the chromosomes look like in the top right panel. And then they were able to take those chromosomes and sequence them into the exact code based on those 4 letters that I mentioned. You can see at the bottom right corner. And this has been published, and it's in databases. It's called the Human Genome Project. Now, is this how they sequence the genome of viruses? Well, no, it is not. So how do we sequence an in silico genome? So the most important point to tell you about how they do genome sequencing of viruses is that they do not follow the procedure that they did for humans. In other words, they do not start with the virus and then take the genetic material out of the virus to sequence. So I can tell you that scientifically speaking, not doing it that way, there's no other way that could possibly exist to get the sequence, the full genome sequence of an organism. So the most important thing is that they're not actually taking this genetic material directly from a virus. So I don't really need to even explain this part, but I think that it does teach us quite a bit to see how these scientists, what they're actually doing, and then saying that it's equivalent to a genome the way that they did the human genome. So this is an overall view of the procedure of this process, and it's the overall procedure is called next generation sequencing, although sometimes that only refers to the sequencing portions. But you can see this is, from a commercial kit, because this is big business. There are companies that sell all of the machinery and reagents to do this process in its entirety. And they have very good literature. And this is one of their flow charts, which describes the procedure. And you see that it starts with clinical samples on the top. And let's follow the left tract here where it says ultracentrifugation, density gradient, and then pure virus particles. So that's the appropriate way to do the sequencing. So in other words, you purify out the virus first, and then next it says extract viral nucleic acids. So you're going to basically take those directly from the purified virus. Now, if they use this technique in that manner, it would be proper, but that's not what they do. And if you look at the right side where it says low speed centrifugation, nucleus digestion, and then semi-pure / enriched virus particles. So now this is closer to what they do, but they don't really semi-purify or enrich really at all. They just remove the cellular debris so that all you have is RNA and other molecules in the solution. But they do not purify out, and you actually have genetic material from probably at least around 100 different organisms. Okay, now the rest of the process essentially makes a library and amplifies those sequences so that they can actually undergo sequencing where you could tell what the specific letter codes is. And then it talks about here random library preparation and sequencing. That's what I just discovered. And it has this one little box here, data collection, storage, processing, and analysis. And that's really a big black box of the real in silico computer procedure that I'm going to get into in just a moment. So let's go on. So this is the paper that was published last March where they did the first genome sequencing of this alleged SARS-CoV-2 virus. And that's what I'm going to describe the procedures from this paper. Now, after this, all the other sequences that were done were compared against this one. So this one is done different from every other sequence that came subsequently. Okay, so this is essentially the procedure. Now, you'd think if they were going to sequence the genome of a virus, they might, and especially given, as Tom described, that the virus particles are in such a tiny amount in the fluid, you would think they would do it from more than one person. But actually, this comes from just one single individual who had respiratory symptoms consistent with an atypical pneumonia and then had a meaningless PCR test result positive for COVID-19. So they took the lung fluid from just that one single individual and they extracted the RNA, and that includes the RNA from every single organism that was present in that lung fluid. So bacteria, fungi, and human cells, and then any other contaminants that might be in there. Then they looked at that RNA and they said, you know, how long are these strands and how much do we have? And then they made what's called a library, a cDNA library or an RNA library, depending on which paper you're looking at. And all that is just making it easy to work with. Okay, so in other words, putting them in folders, putting some little tab labels on them, that kind of thing, so they're easy to work with. And then they do the actual sequencing, and they can do it the power of this technique is that they don't have to sequence one thing at a time and start at one end and go all the way to the other end. They can actually sequence, hundreds of thousands, I think, of different strands simultaneously. So do them in parallel rather than in sequence. And this is why this technique is so popular and so commercially successful, because at the time they did the human genome, it took years doing it the long way. And now they can do it really in a matter of days. So once we have all those strands sequenced, and they can only do short ones. So in other words, if in this lung fluid, there might be some really long pieces of RNA, and they could actually really represent a long sequence. But those are just tossed aside because they can't be used in this high-speed sequencing technique called next generation sequencing, where it's done in a two-dimensional array. So they only have the short strands, and then they put those strands that they've now sequenced. So they actually sequence these 150 base pairs or less strands, and they just remove some of them, or which in a process that they call trimming, but they don't specify how they do this at all. And this is done in a computer. So this is the kind of initial cleaning up the data. And then how many actual reads, and a read is one individual piece of RNA, do they have now? Well, in this case, they had over 56.5 million different separate pieces of RNA from who knows how many organisms. Okay. So how do they go from having 56.5 million little pieces to getting a whole genome? It's a big question. So what I'm going to do is I am going to put you through a little bit of a story here to help you understand how one might go about doing this. And this is going to be related to books and libraries, since we're using those words already. Okay? So everybody ready? Pay attention now. Sequencing a viral genome is like reading a book. The letters in the book make up words, sentences, and paragraphs, just like the letters of DNA make up promoters, genes, and chromosomes. The genome of the book is simply the entire sequence of letters. If you have the book, it's just a matter of reading it. But what if you don't have the book and aren't even sure if the book exists? Can you read that book anyway? Well, this is the situation with SARS-CoV-2. So what do they actually do when they claim to have an in silico, meaning computer-generated, genome? All right, imagine you were looking for a rare science fiction book from the 20th century that was only rumored to exist. So you went to the library and asked for help, and the librarian brought you to a room in the basement, which is pictured here, where you have 116 different science fiction titles, and there are multiple copies of each book. But unfortunately, the books are not in very good condition. In fact, they're torn to shreds. But the librarian reassures you that all of the pages are there because no one has disrupted the room since the books were stored there. And guess how many pieces of paper you're going to have to go through? 56.5 million pieces of paper. So you decide to try and reconstruct the book with the hopes that you'd find the lost book. So you match up the little bits of paper by overlapping the letters, as shown in this slide. You tape them together into longer and longer pieces. You realize not all the matches make perfect sense, and many of the fragments can be matched to more than one other piece. So you do the best you can, and you make multiple different versions using up every single bit of paper. So let's examine some of the sentences that we constructed. So here we use the overlap of the two words writing in, which represents a string of 9 letters. Okay, so this is like a 9-letter overlap. So we have constructed 3 separate sentences here from the bits of paper, and I'll tell you these are all from real science fiction books, and I will reveal exactly where they come from on the next slide. So here's the first sentence. I was at home at that hour and writing in kindergarten and 1st grade. Now, this sentence actually is grammatically correct, but it does not exactly make sense. Okay. Second sentence we have here. The teachers of our country have to be taught to start teaching, reading, and writing in a foolish, facetious tone that the perfection of mechanical appliances must ultimately supersede limbs. ... Once again, grammatically correct, but doesn't exactly make sense. One more sentence. For a moment, he was seized by a kind of hysteria. He began writing in my study, and although my French windows faced towards Ottershaw and the blind was up, I saw nothing of it. Once again, grammatically correct, but not exactly very meaningful. So are any of these sentences correct? Well, let's find out. In the top example, we have the first half of the sentence from H.G. Wells' The War of the Worlds, and the second half from Ray Bradbury's Fahrenheit 451. So we have mixed up these and even included, appropriately so, George Orwell's 1984. So you see that we could have made a lot of different combinations based on this overlap here that were incorrect, in addition to making the correct ones. And this is just using 3 books with a simple example. But having 100 books with 56.5 million pieces of paper makes it even more complex. So after months of matching up little bits of paper, you construct many contiguous strings of letters and call them contigs for short. You finally get over 1 million taped up pieces. Many are long enough to be a book. Do you think one of these might possibly be the book in question? How could you know which one? How could you be sure if you never checked it against the actual book? You decide to pick the longest one and call it a day. Congratulations on completing your task. You've found the lost unknown book, or have you? So that is really how things go with this experiment. And now I'm going to point out some of the data that is actually described in the paper, and it matches up with those steps I just described in my hypothetical story. So in order to match up the little bits by the overlapping letters, they actually used not one, but 2 different computer programs. One is called MegaHit, which really sounds like it should be playing rock songs, and the other one is Trinity. And you can see that these generated almost 2 million different possible contigs, okay? Now, I found it quite interesting that the Trinity program, which generated over 1.3 million different combinations, didn't have any that were very long. They were only, right, just under 12,000. Now, they tell us in advance that they think these virus genomes are about 30,000 in length. But, you know, that number is not based on anything meaningful. We know from looking at genes in humans and other mammals that there's actually quite a wide range of the size of the genes, anywhere from just a few hundred up to 200,000. So it could really be anywhere in there. And if we have an unknown organism, it's amazing that we can know so much about it without knowing anything about it. So what they did was, since it would be really difficult to choose between these 1.7 million combination, they just picked the longest one. Okay, that's pretty convenient. Makes you wonder why they had to generate all these other ones. However, that one that they picked, when they ran it through a database, it turned out it was 89% similar to a bat SARS-like coronavirus. This is kind of, I don't know if the bat thing came from this database or they only looked for the bat virus in this database and not looked for anything else. But 89% is not all that similar. For example, humans are 98% similar to chimpanzees. So while we're not very much like chimpanzees in real life, and here we have a much less degree of similarity. So they also said the whole genome was actually shorter than this contig. So what does that mean? That the other ones the computer added on were just no good and they just chopped them off? So how do they know if those ones were no good? How do they know these were any good? Right? Now, it's also interesting to know that, going back to the book analogy, that this one contig that they chose was made from 123,613 different pieces of paper that were taped together into this one that was 30,000 long. So you see that it overlapped quite a bit because there were only about a quarter of the total represented. So there must have been some kind of like a big overlap somewhere. Okay. So how do they know which of the reads were correct? How, you know, the contig, sorry. How do they know which that this genome is real at all? How do they know where these sequences came from, what organism, since they came from a mixture of organisms? How many errors are introduced in each step of the progress with each different computer program that's used? And of course, any experiment, in order to be scientifically valid, has to be able to be replicated. So can this experiment be replicated? So just to review, the difference between a real genome sequencing comes from an intact, isolated, and characterized organism, can be reproduced without error, and represents the actual code of an actual organism. Whereas the in silico genome comes from a mixture of unknown sources, cannot be reproduced without error or the showing of false mutations, and represents a theoretical code of a theoretical organism. So what are variants? Well, variants are essentially attempts to repeat this experiment. And what they are doing when they repeat this is slightly different, because instead of templating it against that bat virus, now they're actually templating it against the results of that study. So the sequence that was published as a result of that study I just described now is the standard sequence, or considered the standard sequence, for the SARS-CoV-2 imaginary virus. So since that time, when anyone else in the world wanted to try to do a similar sequencing from taking the fluid from just one patient, they would essentially follow the same protocol, except at the end, compare it to the results now of this genome, the new one. And in fact, there were companies making the equipment for this that published exact procedures on how to do this. Okay, so everybody is doing this pretty much exactly the same way, no matter where they are. But are they getting the same results? Of course not, because they're not taking the genetic material from a pure sample that would have the same sequence time after time. They're not going to be able to get the same results. So each time the results are slightly different. They might only be off by a fraction of a percent, but they're different. They cannot get the same results. Now, what they do is they call this variance. But what this really is an inability to validate or replicate an experiment. And in other words, it invalidates the results of the initial experiment. But they've really capitalized on this, and you can see, and this is not a new slide, this is a couple of months old, but I've heard, in the thousands, like well over 5,000 different variants or genetic mutations have been reported. And this is very dangerous, because we have the impression that a genetic mutant of a virus could potentially even more dangerous than the original virus. So this is essentially a fear-generating device, because each one of those dots that you see on there would generate fear. This is a real journal, by the way. This is the journal where they publish the research that's considered for the Ig Nobel Prizes, if anyone wants to look into scientific satire. So I want to relate this genome sequencing to the vaccine, because this is what I think is really the new paradigm of vaccines and viruses. Okay, so what we have here is that the results of the genome sequencing that I just described, that's completely theoretical and imaginary, what they can do is say that some of those sequence letters in there belong to a gene for this or that. And they have characterized one, especially that you've heard a lot about, which is this so-called spike protein gene. And from that imaginary genome, they pull out the letter sequence that will make in a recombinant organism some kind of protein, but it doesn't come from a virus. And what they're doing is taking that sequence and plugging it in a ready-made platform that can be used to manufacture a new experimental vaccine in a matter of hours. So what it is that companies like Moderna who have really pioneered this, but as we see, the other big pharmaceutical companies have gotten in, that they have a delivery vehicle for genes into our bodies. And in some cases, it's a lipid nanoparticle. And in some cases, it's adapted from another alleged virus or some other kind of particle, as Tom described with the Johnson & Johnson experimental injection containing the envelope from the adenovirus. But all they need, because all of these sequences of RNA can be made by a machine where they just simply has a computer terminal attached to it, and you type in the letters of the sequence, or they could probably just be imported directly from a digital file. And then you click on and make sure you fill all the reagents in the machine, and it will make strands of DNA and RNA. So all they need is that computer file from the in silico genome to be electronically sent, and they can put it right in the synthesizer, and basically in a matter of hours or a day or two, they just put that gene sequence into the vaccine genetic platform, and then boom, you have a brand new vaccine. And this is how they were able to roll these products out in a matter of months, rather than the minimum 10 years it takes to make the previous generation of vaccines using the tissue culture experiments like Tom and I described earlier. So if we kind of review the pandemic recipe from the past or the present, not the future, is that you need to do the tissue culture experiments, which we explained have been disproven, but that was the prior proof of virus isolation. And then they would use that same tissue culture to develop the vaccine, because the fluid from that tissue culture is used to make the vaccine. And then that took more than 10 years, as I mentioned. And now we have the next generation where we have, and I believe in the future, they won't even do these tissue culture experiments. They'll just go right to the genome experiment, which is even easier and quicker to do. So they'll publish an in silico genome, and then they will use that to very, very quickly make a new vaccine. And rather than getting approval, which would require them to prove that this injection is safe and effective, they will use the emergency use authorization, just as they have done in the current pandemic situation. So they would basically create a fake virus very quickly, show a fake genome, use that as a pretext for a pandemic, and then use the emergency authorization to bypass the approval and clinical testing syndrome, so that you can essentially create this whole operation with the vaccine in less than 12 months, as we've seen play out. So just to review, a genome is the full code of an organism. An in silico genome is a theoretical construct. And the new paradigm of virus pandemic creation involves mimicking the symptoms of a known disease, perform the tissue culture, quote unquote, isolation experiment, which I believe will be obsolete in the future, create an in silico genome, develop a test like a PCR based on the in silico genome, and manufacture the gene therapy vaccine based on the same genome and get emergency authorization. And that's what we've just experienced, and I believe will be the continued paradigm going forward. And that's the end of my talk about the genetics, Tom. I know that's super exciting stuff here.
That's a mouthful.
It certainly is. I hope that everyone is still awake and ready for some Q&As. Leah, you ready to drive the car?
Yes, I am. I just want to get a quick check from everybody out there. How's my microphone sounding? I've turned it up a bit. Can everybody hear me? Okay, great. Okay, so we do have some really good questions. A lot of people are asking about India. There seem to be a lot of people dying in India. Some people are saying it's happening because the vaccines were rolled out. Do either of you have any insight on what's happening in India?
Well, I think there's, first of all, a lot of misinformation. For example, I know that they were using video footage from a chemical leak several years ago, saying that now people were dropping dead of COVID using the same video footage. So it's very, very difficult to tell if there's really anything going on. But if there are people who are getting sick and dying, we know that it can't possibly be from a virus. So we would have to look for other causes.
Okay, great. Thank you. Also, people want to hear about shedding. The idea is that somebody's going to go get a vaccine and they're going to walk around and they're going to shed and we're all going to get something. Could you speak to that, please?
Yeah, I'm going to let Tom go for that
No,
one.
I'm going to let you talk. I'm hoping you talk about that. I talked about that yesterday, but go ahead.
Your mic, Andy, you just muted.
Sorry, that was an accident. But yeah, I think we probably both need to talk about this, but I'd be happy to start off. I want to say that, one of the things that I've been very, very careful about, and I've been really critical about the whole pandemic, is that the government and the scientific establishments have really jumped on things very, very quickly without any evidence, and played things up and made decisions based on that. And I don't want to make the same mistake. So with respect to this, because there are bad consequences, I can see that a lot of people are calling for the separation of vaccinated and unvaccinated people, like telling us to avoid them, stay away from them, quarantine them, et cetera, et cetera. And this is a divide and conquer strategy that, could really be devastating. And before we would recommend that anyone separates or divides from anyone else, I would want to be very, very sure about what's going on. So there have been, so far, what I could tell, a lot of rumor and anecdote. So in other words, people making various reports, like through email, maybe to someone who's speaking out, that, I think, I'm harmed by this, by someone who's been vaccinated, et cetera. And I've come across some people that have approached me individually with their stories about what's going on. And there was no evidence in any of the people that approached me that would said that there was a relationship here. So if people are serious about this, someone should simply start looking at this like in a scientific manner. So if there are 1,000 people who have experienced this, let's do a survey and let's see, what exactly is their exposure to people with vaccines? What exactly symptoms did they experience? What were the timing of things? Because this is epidemiologic evidence. And this is where we start only to generate hypotheses. It's not where we understand things. So we need to get to that where we can actually generate hypotheses. Because right now we don't really have anything except fear and rumor. So I wanted to just say one more thing about it, which is I've thought about, well, if it were possible, what would be the mechanism, that we would transfer something? And I've heard, several things proposed, but I think most doctors are who are talking about this are saying that it's the spike protein, so that the genetic vaccine causes the recipient to manufacture the spike protein, and then maybe it comes out in some body fluids or some way gets into other people's bodies who are around them. And I'd like to point out that there's no model for this that I'm aware of happening. So our bodies do make lots of proteins. In fact, humans make about 100,000 different proteins. And many of these are in our bodily secretions, like in our tears and our saliva, for example, are antibodies. Now, we know about antibodies, right? It's a pretty common thing. And we think antibodies, are involved in autoimmune diseases and in other diseases. But it doesn't happen that, like you kiss someone and your antibody from your saliva gets in their mouth. It doesn't like start attacking their mouth, right? That doesn't happen. And there's no other proteins that we know of that leave one person and then perform an active function in another person. And this is extremely unlikely because proteins, once they get into the environment, they generally become quickly denatured. In other words, they need to be in a very, very highly specialized biological environment for the most part, with structured water and the right electrolytes and the right this and that, in order to be in the right shape to do their thing, whatever that is, whether they catalyze a chemical reaction or they, bind oxygen or whatever they do. And once it gets outside of your body, chances are it's going to be quickly denatured. It won't have that microenvironment or be able to do anything. So I think it would be, extremely far-fetched. But there is one way that I think people definitely could be affected, and that is simply through empathy. So if you have a loved one who you're worried about and they're getting injected and you are worried about their health, and then you have a psychosomatic reaction, like that certainly is possible. And really that shows the amount of compassion and feeling that you have for that person. But let's not construe that with some, physical danger of being near vaccinated people. So Tom, I want your impression too, though.
Yeah.
That this is everyone wants to ask us this.
Yeah, right. The other thing is that I don't know anymore really about what's happening in India, because it's hard to believe much of anything. But the only thing that I do know, and this is because I did an interview with a man named Indra Singh, who is, I think, probably the main person who's chronicling the so-called farmers' revolt in New Delhi. And he came on an interview with me, and I would encourage everybody to watch that interview on one of my podcasts. And basically, he described that there were, essentially the government in India is trying to get rid of small farmers. No surprise, because that's happening all over the world. And in India, the farmers are deciding they're not going to take that. And so literally millions of them descended on New Delhi in a sort of march of protest. And they have a lot of support, and they organized food and water and getting rid of waste. And what was so interesting about it is there's millions of these farmers, he said, living in close quarters, sometimes 10 sleeping in a tent. And up till then, and this was maybe a month or so ago, there had no evidence of any kind of COVID disease. And they were very well organized. In fact, it was very inspirational to hear that these essentially poor people were decided that they're going to fight for their lives and their land and their livelihood and their way of life. And they had support from the town and the surrounding people with food and water. And then as soon as they were sort of gaining traction, then you see this sort of COVID situation hit. So that makes me very suspicious that there could be some way that there's somehow targeting these people. I don't know if it's through injections or some other toxin or some sort of electromagnetic field exposure or something. I don't have any evidence for that, but it's worth exploring. Which brings up my point about the thing I wanted to say about shedding is, clearly we, and I get a lot of stories, and I talk to other doctors who share stories of people who've gotten sick following what they seem to think is an exposure to somebody who's recently given an injection. And what I see, similar to what Andy says, is this is a public health catastrophe, right? If there's a situation, remember the role of epidemiology, which is what these observations are, We don't know whether they're true or not, whether they're caused by what we think they are or not. But the role of epidemiology is to say, hey, there's something happening here. We should look into this. So there should be a proper, formal, epidemiological investigation. I'm not sure exactly how they would do that, but I'm sure it could be done to see whether or not people are getting sick from exposure to others who've been injected. And that would be the first step. So yes or no. But amazingly and predictably, I would say, that never happens. It just, there's no investigation. There's just name calling on both sides. Yes, it's happening. No, it isn't. Nobody ever gets to the bottom of it. And if they found that it was happening, then, they should do some sort of experiment to find whether there's some sort of mechanism that could happen. I know that Andy sent me a study that showed that, radioactively tagged lipid nanoparticles are excreted in the urine and feces. And so that was after one intravenous injection. So, that's a possibility. But the first step is we don't have a scientific procedure in this country so that these things can be investigated. And so just like everything, it just becomes an argument based on nothing. Nobody ever gets to the bottom of anything. Nobody ever does clear studies that you could say, this is real or this isn't real. And so it just fades off into the Netherlands of yet another unsubstantiated rumor. Unfortunately, this unsubstantiated rumor, and I don't know whether it's real or not, serves to divide people, as Andy said. And so now we have yet another way that we can say, I'm not going to be around this person. And so we have now even more divided. And that's a very dangerous step to take. And so I'm not willing to go down that road myself until there's very clear evidence that this is what's happening. But I do think we should look into it. That's the role of science. That's why we spend trillions of dollars for a public health system in this country. Maybe they should do something for a change. And in the meantime, let me say one more thing. I am perfectly willing to say until this thing is put to rest, I am willing to say we should have a moratorium on giving any further injections until the issue of viral shedding is proven one way or another. I am willing to do that because what can it hurt?
Thank you. We have a couple of guests who would like to ask questions. Teresa, would you please unmute yourself?
How do I do that?
You did it. Hi.
Okay, hi. Thank you. Thanks for all these great informative videos and so forth. I do, to my chagrin, have family members who do not agree with me on the basic premise that there is no virus, and other people I speak to, and I do a logical question that comes up is, as you just said, you said, okay, we do not have evidence that this so-called pathogenic effect is caused by a virus because it's also produced by, a non-virus, saline, what have you. I agree that does not prove that the virus caused it, but I wouldn't say it proves that there is no pathogenic element. And so when people say, for example, you yourself said the bacteriophages have been isolated, in fact, purified and isolated. Why and how did they do that? And why and how, I'm sure you can't say why, but did they not do it with this so-called virus? So that's question A. Do we or don't we have actual purified, isolated things in that nanometer range that can be called viruses? We're taught that doctors didn't wash their hands and women in childbirth died, and then they started washing their hands and they stopped dying. So what factor was involved there, right? What are pathogens? And the second part of that question is, if we are talking about something like something that's upregulated in the body to cause these presumed symptoms, whether they're pneumonia, whether it's neural, whether it's, epithelium, kidney, I don't know what else they're claiming in this vague constellation of symptoms is supposedly COVID. If this is in fact related to EMF, what would you think is the upregulated factor and mechanism that is in operation there?
Okay, thank you, Teresa.
There's a lot of questions there. I'm not sure I even remember them all. But the first one that I remember, so why, if we can isolate things of that size, morphology, and complexity, why don't they do that with viruses, pathogenic viruses? And I just want to point out again that they don't do it is not in dispute. Everybody agrees with that. They haven't done it. There's no reported studies showing they've done it. They say that there's not enough to find. I've also heard the explanation that viruses are intracellular pathogens, so you can't find them outside of the cell. And then I asked one of them, well, how does it get from one person to another? And they said, well, it buds out the cell and then it goes to the next person. And I said, why don't you find it then? And that was the end of the conversation. So the reason they don't find it like that, now they don't look, but previously because they couldn't find it, period. So they went to the tissue culture experiment as a way to prove it. And as we now know, that doesn't prove it because if you do the same experiment without adding something, you get the same cytopathic effect. Now, because they don't purify anything, you could get different results, whether you, take the secretions from somebody with measles and it has all sorts of toxins in it and, other pieces of all kinds of things. And so you may get a different result from the culture, but that doesn't prove it's a virus. We have to use logic. If you want to say it's a virus, this is why I use the ping pong ball example. If you want to say that the ping pong ball knocks down the wall, you don't put it in a bucket of stones and throw it at the wall. Every human being has the sense, if that's what your claim is, and mind you, we're locking up the whole world based on this claim, you have to take only the ping pong ball, throw it at the wall, and see what happens. You can't use a computer simulation. Nobody believes that. You can't take the advice of 10 experts. Nobody would believe that. I thought of another example. If you have a firing squad and you have 3 people with guns and 3 people who sing the Star Spangled Banner, and you have a prisoner and you say, ready, set, fire, and 3 people with guns shoot the prisoner and the other 3 people sing the Star Spangled Banner, and you say, which caused them to die, the guns or the Star Spangled Banner? Every human being would say, okay, we'll do one where just the guns and the other people sing the Star Spangled Banner and we'll find out. But we don't do that anymore. We just argue about which one it is. And that's not the way to find out the answer to anything. Now, as far as the question of what happened with doctors, washing their hands and then not creating, childbirth fever, it's the same problem. So here we had a situation where doctors would go from doing autopsies, and obviously you do an autopsy on dead people with formaldehyde and all sorts of chemicals, and the person died and their hands were all bloody and full of chemicals. And then they would go right from there into the delivery room and stick their hands up a delivering woman's cervix into an open uterus. And then sometimes something bad would happen and that woman would get sick. And then they said, you see, that was caused by a bacteria. I think that's what they said because they didn't know about viruses then. There is no justification for concluding that was from a bacteria. there is certainly justification of saying, hey, buddy, if you're going to go to do an autopsy and put your hands in formaldehyde and have blood, I mean, there is such a thing as like serum sickness. If you put blood into somebody else's body that's untyped and full of God knows what, you might make somebody else sick. So don't do that. Like, wash your hands so you're not putting blood and formaldehyde into somebody's open uterus. If you want to say, I mean, if you're going to be rational and scientific and logical and conclude that was a bacteria, the only way to do that is to say, okay, take the bacteria from that person and insert that into the open cervix and see if they get sick. And as far as I know, that never happened. And this is the problem with people. They confuse what we're saying as, so you mean people didn't get sick? And of course we don't mean that people didn't get sick. They obviously got really sick. And of course we think you should wash your hands of blood and chemicals before you stick your hands in somebody's cervix. That's not the issue. Of course people get sick. They get sick from all kinds of reasons, including starvation and wearing masks and electromagnetic poisoning and drinking bad food or bad water and eating bad food and all kind of toxins in the air and the water and glyphosate and maybe cyanide and who knows what. The reason it's so hard to say, well, it's because of this, is the same thing. Everybody is so wedded to the virus theory that there's no actual research in looking into anything else. And so we'll go, hundreds of years without finding out, oh yeah, if we had in a functioning public health establishment, they would do proper epidemiology, generate every hypothesis they can. Is it starvation? Is it, stuff, nanoparticles being sprayed in the air? Is it 4G wavelengths? Is it 5G wavelengths? Let's find out. I mean, what's the problem? Let's find out. Let's do real science and have an actual culture and a scientific establishment that works.
Very good. Dr. Andy, what are your thoughts?
Well, I really, Tom gave a fantastic response. I just want to add one very minor point, and it has to do with the hand washing issue. And this, is really a bit of common sense. But, when you're doing an autopsy on a dead body, right? When flesh is dead, it changes. It starts to get consumed by microorganisms, and this is the natural saprophytic process in nature, which reduces the flesh back down into the constituent components so that it can be part of the earth again. And whenever you have microorganisms consuming rotten or dead flesh, they're going to generate waste products which can be toxic. And this is what food poisoning is. And this is also probably the most likely thing that caused puerperal fever, which is the childbirth fever that we're talking about here. Because, so the bacteria is growing in the dead body, or it's growing in the old, steak that was left out. And it makes waste products that are toxic, just like our own waste products are toxic. If we took human feces and inserted it in the birth canal, we would also cause illness. So we're essentially taking these toxins from the waste product of the microorganisms and putting them in the subject. And the same thing happens, when we leave food out too long, like at the summer picnic, and there's mayonnaise that in some cases, the Staphylococcus bacteria will eat that spoiled food, and then it will create what's called the staph toxin. And then if we eat that toxin, not the bacteria, but the toxin, poisons our intestines and causes a diarrhea reaction to flush it out. And it's very, very important not to confuse that with a bacterial infection, where the bacteria would actually invade the body and grow and cause disease. And when experiments have been done where just the pure bacteria has been put in a person, there's been no ability to reproduce the disease.
I'd like to actually add one thing, which I forgot to say about this idea that like a protein, like for instance, in this case, a spike protein can cause disease by being expelled in the air and cause another person to be sick. I don't know. Personally, I don't know any evidence that anything like that actually happens. You know, when you think of, it's not that toxic proteins, sometimes enzymes, can't make other organisms sick. You know, a classic example is snake venom, which I believe is some sort of enzyme toxin. But you have to realize that the snake goes to great lengths to have a sort of delivery device that makes that toxin poisonous. They don't just spray it in the air. At least as far as I know, there's no snake that sprays toxins in the air and makes people sick. They have a whole system of fangs and probably, you know, it probably once they bite you, then something gets changed and liberated, and then it becomes an active enzyme or protein that makes you sick. And I think the same with all sort of biting insects. So that's why it's very unlikely that it's a pure protein that somehow is making other people sick. But again, we do have a kind of novel delivery device that is a lipid nanoparticle. And, you know, as I said, that should be investigated as to whether that can, you know, remain intact and deliver some sort of thing that can make somebody else sick. I'm not saying it does or it doesn't. What I'm saying is that should be the role of science and the public health establishment, to never roll out some injectable device where that exact question hasn't been thoroughly answered.
Yeah, that's good. I have a question, and I'm getting feedback on somebody's microphone. Okay, thank you. So from Christina Beard, with regards to the transhuman and mind control implications of this vaccine, do you think there is any hope for those who have already received it? At face value, the prognosis seems grim, but the field of epigenetics has shown the power of our mind's ability to alter the expression of our genes. I would love to hear your thoughts on this and whether it could apply to those who have already been injected, especially for those who may have been injected against their will, such as children or adolescents. What's happening to people who have had it, and what can be done to help them?
Well, hopefully there shouldn't be any children who have received these injections so far. You know, I think that there's really no way for us to know. You know, I've heard people make predictions that, you know, at some point something's going to happen, or it's going to interact with some other factor, like that there'll be a signal like a remote control, or maybe it's through 5G installation. But really, you know, there's nothing disclosed that would help us figure this out. And in terms of the safety studies, they're practically non-existent. Normally, we would have at least, you know, a year's worth of safety data at the time a vaccine gets approved or something like that. And that's the only way to know what are going to be the long-term outcomes. So I think that what I would, you know, say just as casual advice to people who have been, you know, received this injection and realized that was not a good decision, they should just try to do the best they can to maintain health, in, you know, physically, psychologically, and spiritually. And then they'll be the most resilient they could possibly be to any deleterious effects.
Thank you. Okay,
sorry. Let me add to that. So I completely agree. There's no way either of us or anybody right now, as far as I can see, can answer that question as to what is actually going to happen, because we've never been down this road. But I would say that if there's any good news, it's that the whole idea of stabilizing an RNA so that it will continually make a certain protein. The good news is it fundamentally goes against biology, because as far as I understand, mRNA lasts like literally nanoseconds. And so basically, if you were just to inject pieces of mRNA into somebody, it wouldn't do anything because it would be degraded within literally seconds, and nothing would happen. There would be no protein made. It wouldn't get turned into DNA or anything. So the entire gambit here has been to somehow work against nature and get this piece of mRNA to be, intact and functional for a really long time, maybe indefinitely. And I'm not sure that that's so technologically easy. Like, I know they have these lipid nanoparticles and they put them in vectors and they have PEG and they have all kinds of techniques of doing that, and maybe putting in, I don't know what other kind of nanoparticles, because they don't really tell you what's in them. So I have no doubt that that is what may be attempting to do. But the good news is, we're still human beings who we decide. And when I say we, I don't exactly know which part of we or us is deciding. But I have a strong belief that the chemistry, the physical stuff follows consciousness, not the other way around. And so when you speak about epigenetics, that's the sort of part of us that is really in control, which modern science has no concept of at all. And I don't think you can hurt that part with an injection. Now, that part may not be able to overcome a very strong anti-biological assault, and that is what I understand to be the transhumanistic agenda. But I don't think it's so easy. So that, I think, it works in our favor.
Very good. Thank you. So now we have a few guests lined up. Please ask one question. We're going to try to get as many more people in as we can, and so we have a little more time to answer. Thomas, please unmute yourself. And just, I believe it's on the top. You can just hover your cursor around.
Better?
Yes.
Awesome. Hi,
Yes.
Tom, Andrew, can you hear me?
Yes.
Yes, great. Hi, thanks for having me. What I want to, thanks for the lecture. I really appreciate it. And as a return, I actually teach a course in genomic sequencing in the fall that I welcome you both to take, and we can clear up some of the issues that you might have had in terms of that black box, which is not a black box to people who know bioinformatics and sequencing. Just wanted to share with you this image here, which you can see is the supposed, SARS-CoV-2 viruses and the coproteins. These are two independent lines of evidence because they ran a Western gel which detects the protein, as you can see, correct? So this coprotein here, these are actually predicted in the genome itself. So how does that work with your model if we have basically three independent lines of evidence that the genomic sequencing is predicting the spike protein produces a specific type of amino acid sequence that we can visualize with this cryo-EM and also detect on a Western blot, which detects the protein sequence?
Hello, everyone. On our recent COVID Myths webinar with Dr. Tom Cowan, we were asked a question by a participant named Thomas about a paper where he claimed that the virus had been purified. At the time, we were unfamiliar with this paper and were unable to answer. So in the last 2 days, I have gone and looked at this paper and discussed it with Dr. Cowan. And this is our answer to the question. So I'm going to share my screen so we can look at this paper together. So this is the paper of interest called In Situ Structural Analysis of SARS-CoV-2 Spike Reveals Flexibility Mediated by 3 Hinges. So the participant had claimed that in this paper, the virus had been purified and that the genome was constructed by a nanopore analysis, which is a way of getting a complete strand of genome and sequencing it one base at a time, very different from what I described in the presentation. And so I looked at this article, and these are the pictures that were alleged to show purified virus particles, and they're done by a technology that I've never heard of before, which is called, sorry, it has a very fancy name. What is it? Cryo-electron tomography. So I guess sort of combining electron microscopy and computerized tomography like in a CT or CAT scan. So as I have said many times, the most important thing when you're looking at an article is to look at the methods section, because if you don't know how the experiment was done, you would not be able to describe or understand the results. So I'm going to stop sharing for a minute so that I can bring up the other section of the paper, because interestingly, the paper that I just showed you has no methods section whatsoever. And if you go on the website, what you'll see is in a little space over on the right side panel, there is a link to supplementary materials, and you have to click that link in order to find the methods. And interestingly, the article itself is only 6 pages, but the supplement containing the materials and methods is 25 pages long. But I think not too many people probably look at this. And so here now I'm sharing the screen again. We have the supplementary materials for this paper. You can see the same name of the paper. And what's included in here is the materials and methods, which is the part that I'm going to be discussing, and then figures and tables and some other material. So if we go right here to the beginning, we can see in this first section, SARS-CoV-2 propagation. It says SARS-CoV-2, and it says isolate MUC-1, a kind gift of G. Dobler, the Bundeswehr Institute for Microbiology. Now you'll notice there's no reference listed here, and none of the references are publications about this isolate MUC1. So I went and tried to search on PubMed and in a general search engine for this isolate using G. Dobler's name. And then I went to the Bundeswehr Institute for Microbiology website to try to find it. And there was no information whatsoever about what this isolate is, where it came from, or anything like that. So this is obviously a big problem. Without knowing this, we don't know what it is. Since I'm unaware of any examples where a virus was purified directly from lung fluid or some other clinical sample, I'm going to assume that this is a tissue culture supernatant, which in other words means that they added lung fluid to a tissue culture under some unknown conditions, but probably with increased antibiotics and decreased nutrition, and then showed cytopathic effects, and then did some kind of perhaps genetic testing like a PCR to show that to make a claim, that the virus is present, and then they put this in a test tube and give it for this experiment. But we really, can't evaluate the experiment at all without knowing how this isolate was prepared and what exactly it is. So we really don't have to go any further, but I certainly will go further anyway, just to be thorough. So the next section here is the virus preparation for the electron microscopy. And we see that it says here that the identity of the virus was verified by Sanger sequencing of SARS-CoV-S gene fragment spanning the furin cleavage site. So this is Sanger sequencing is the older method of doing sequencing where you can have a whole piece of a genome or a whole gene and take one base off at a time, so different from the next generation sequencing where everything's done in parallel. And this is, definitely a time-honored way, but here they're only doing it for a very short segment or fragment of a gene. So in other words, a very short strand of RNA. And we have no idea what the origin of this is. Now they're saying that because of the presence of this sequence, that means the virus is present. But there's no validation study ever showing that to be the case. In other words, they never isolated or purified a virus and then did this type of a test on it to show that it can only come from there and not from another sample. So there's no control experiment, etc. And these came from infected viro cells. Okay, now they did say that they subtracted out the sequences from the viro cells, but there could be other sources of genetic material in there as well. So this doesn't really tell us very much. And then in the bottom part of the paragraph, it describes how the cells were harvested. Now, this is where Thomas claimed that they were purified by density gradient ultracentrifugation. However, if you read closely, here's what it says. For virus purification, viro cells were infected at MOI of 0.1, the supernatant harvested at 6 hours post-infection. So that means that the sample MUC1 that was noted above was added to a culture of monkey kidney cells, and they were left for 6 hours. And then they added paraformaldehyde, which killed everything in the culture. Then they took the fluid out of that culture. So they left the cells behind and just took the fluid, and they did low speed centrifuge on it. Okay, so what this did is just move some things to the bottom in a pellet and left some things in the liquid, but it doesn't purify a virus out. Then the clarified supernatant, so just the fluid, so the cellular debris was in the bottom of the tube and removed. The fluid was now placed on top of 20% sucrose cushion in PBS, which is phosphate buffered solution or just salt water, and centrifuged for 90 minutes at 30,000 RPM. Now, this is not a density gradient. A density gradient would have to have different concentrations of sucrose throughout the test tube, with the higher concentrations on the bottom where there's increased density. So in other words, the 20% sucrose is only one density. So this will not separate the viral particles into a band that is isodense or all having the same density. And what they did here is they said, basically, they took the pellet out of that. Okay, so that's different than taking a purified band. So this was not a complete purification process. They were just removing some big debris out of here. So this is definitely not a proper purification, but also remember, even if it were a proper purification, it doesn't come from a sick individual. This comes from a tissue culture. So even if we did purify particles out of there, there would be no way to know where they came from. Okay, now I just want to talk about one last part of this procedure to finish answering the question, then I'll give some responses from Tom. And I don't necessarily want to go through this whole thing, but essentially they follow the same exact procedure for the genome that I described in my presentation earlier. And you can read this more closely if you want to, but here is one of the most important sentences. If I can find it here. Where was it? The reads. Here. The reads longer than 40 base pairs after trimming using Cut Adapt version 2.3 of Universal Illumina adapter were used as input in the guided assembly Galaxy pipeline. Sorry, I missed this before. The library was loaded, blah, 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 and generating 20 million reads, each 150 bases, which should be or less long. And this is comparable to the 56.5 million reads I described in earlier in the presentation. This time they only had 20 million reads. So you can see why is it so different? They have less than half the number from this sample. And these were subtracted from the Vero cell genome, so any Vero cell sequences, and they didn't specify if it's only genes or if it's in the entire genome sequence. They were filtered out, and then any reads between 40 and 150 base pairs long after the trimming process that I described were then put in another software program, VirAmp. And that was run with default parameters, digital normalization option, and the reference genome of the reference beta COVID, blah, blah, blah. This is the bat SARS-like coronavirus, I believe the same one that was used as a template in the article that I mentioned. So essentially it's the same exact procedure that created this theoretical computer-generated genome of a hypothetical virus. So I think you can see from that analysis, we have a feline visitor over my shoulder, that we really can't say that there are any valid conclusions of this article because it did not work with a purified virus, use the same in situ genome sequencing procedures and same tissue culture experiments as all of the other studies that we've described. So I just wanted to read a few comments from Dr. Cowan, who sent me for this video. So number one, could you describe the medical situation of the person from whom the original sample was taken? Well, this is not possible as I described because the MUC1 isolate is not described anywhere that I can find. Dr. Cowan's second question or comment, we are told that the lungs of people with COVID are teeming with SARS-CoV-2. Is this true? And if so, can you give us an estimate of how many SARS-CoV-2 virions would we expect to find in a typical BAL sample? If there are few virions in the patient's lung, then what is the theory of how the virus is making them sick? So in other words, we're told that the virus has to bud out of a cell into a discrete particle in order to, one, go and infect other cells within one person's body who's sick, and then also to spread to other people. So if there aren't enough of those to even measure, then how could they possibly do those other functions? Okay, next question. Is the SARS-CoV-2 the only virus you would expect to find in the person's lungs? We're told that living beings have a, quote, virome consisting of millions or even billions of many, many different types of viruses. Would we not expect to find these in the patient's lung? So in other words, even if you could say that the tissue culture toxicity or cytopathic effects were from something in the person's lung fluid, even if it was from a virus in the person's lung fluid, how could you know what virus was since you started with a mixture of things? Okay, next question or comment. We also know that there are exosomes produced whenever a person is sick. How would you distinguish between an exosome and a pathogenic virus? Well, even the virologists themselves in their own literature have said it's impossible to distinguish these from each other. And this is why they really gave up on finding virus particles in the early 50s before Ender's tissue culture experiment was published. Next, why didn't you do filtration ultracentrifugation directly on the lung fluid? That way, you would do the experiment by inoculating the Vero cells with either purified virus or a combination of viruses, and then also inoculate the Vero cells with the rest of the mixture with all the viruses removed. That way, you'll know which part is causing the CPE in the Vero cells. Well, of course, the principles of experimentation is that you isolate a single variable to test. So that variable should be just a single virus, not a mixture of unknown multitudes of constituents in lung fluid. So excellent point. Also, if you did the filtration ultracentrifugation directly on the BAL fluid and found a virus, you could get the €1.5 million prize put forth by Samuel Eckert for directly isolating SARS-CoV-2 from a sick person. So that would certainly should provide some incentive to do that experiment if it were possible. Next, When you pass the unpurified sample through the viro cells, how many virions approximately do you get? And since there are many, many other viruses present in the VAL fluid, how come these don't culture propagate on the viro cells? Is it only SARS-CoV-2 that replicates? How do you explain this, since if these other viruses were in the lungs, they must also propagate in living tissue, as we are told they do? Excellent point. Next, can we see the entire slides from the cell culture experiment to determine whether any other viruses are propagating besides the one you pointed to? If there are, how do you know the genetic material you're finding didn't come from them? Now, this is an excellent point. We always see these published images, like I showed earlier, that are zoomed in just on one or two particles of interest. However, if we zoomed out and were able to search around everything that is seen on the slide, we would likely see a number of particles of different shapes and sizes that we would say we can't really tell what's what. And lastly, is it possible, since we know that all matter has an energetic imprint or component, that what is being propagated through repeated cycles is the imprint and not a physical toxin? Or do you believe that only physical substance exists? And if so, do you have a paper you can reference that proves this? Now, this is a very interesting topic, and perhaps a little bit beyond the scope of this paper, but it certainly calls into question the principles of homeopathic medicine, and is something that definitely should be looked into, because we know that there are experiments where these characteristics can be propagated in such a manner. Well, thank you so much for listening to this explanation. I hope it enriches the playback experience for our webinar, and I look forward to seeing you again in the future.
Pamela, would you like to ask one question, please?
Yes. Assuming that you have proven that viruses don't really exist, I have wondered, because I knew, Andy, that you attended the summit in California, and you know what I'm talking about?
Do you mean the reunion summit?
Yeah, the reunion summit.
Yeah, I wasn't there in person, but I did participate in the online event.
Right, and I did too, just online. But it really begged the question to me, because I'm convinced now that there really aren't any viruses. I haven't heard from you anything particularly about what you think could be contagious, other than maybe 5G or something emotional, something that passes from person to person at another level. So I've wondered several times and been wanting to ask you, do you think it's possible that the whole world now is very materialistic, and especially modern medicine is very materialistic, that there's something at the level of spirit that can't be measured and can never be seen that could actually be contagious? In other words, that the spirit world could be real and could wreak havoc in us. Have you ever thought of that?
Well, yeah, and it really calls to mind some work done by Jerry Marzinski about schizophrenia, and he has a theory about that, and has actually had some successful interventions with people with schizophrenia based around that kind of conceptualization. So, I think that is kind of a really helpful thing to look at. I don't like to use the word contagion per se, because that really implies that there's like an infectious agent being passed. But there certainly are lots of phenomenons where people seem to spread things to each other, right? Like we spread ideas, for example. There are trends, right? Things go viral that people share interest in the same content. People can spread laughter. In young children in classrooms can often spread vomiting. Women who spend a lot of time together synchronize their menstrual periods. So there are many, many examples where variety of different things that are both concrete and abstract seem to spread from person to person or among people. And the truth is, we really don't know the mechanisms by which these processes occur. We do know that illness is not really spread this way when it has been tested in experimentation, but we don't really know the way this information gets spread, and there are many different possibilities. We do know a little bit about pheromones, that they certainly communicate information, but haven't been shown in this particular type of phenomenon. We know that we generate all kinds of waves, right? Well, acoustic waves, electromagnetic waves from our body. We could be communicating through those vibrational frequencies. And there are many other mechanisms. And I think that if we can put aside, this false germ theory paradigm, which drives almost all of the science with respect to illness in populations, and the idea of illness transmission, then we could probably find out, right, the real mechanisms of some of these things and characterize them better so that we can understand them, and that our understanding will actually fit with our observations.
Okay, great. Thank you. All right. Let's call on Harry. Please unmute yourself.
Hello. So I've heard at least one of you talk about the immune system as being different than what the conventional narrative is. So I'd like to hear both of your perspectives on the immune system. Does it exist? And if so, how does it work? And then Tom, you've talked about ribosomes not even existing. So I'd love to hear your perspective on that.
Thank you, Harry.
Yeah, that's a long story, but so the whole concept of the immune system really came about because of viral infections, a little bit bacterial infections. So therefore, there has to be some way for human beings to ward off these infections, and so therefore there was invented the concept of an immune system. If the other way to look at this, which is what I've been saying, is a much more realistic model, is we have various detoxification systems, starting with bowels, which happens through the liver, and then through the urine, and then through sweating. And then if we still have the buildup of toxic material, then the bacteria come to help us out by essentially eating dead and dying tissue. And unfortunately, we call that a bacterial infection. And then when the tissues break down, they break down into particles, which we unfortunately call pathogenic viruses. And as far as the replication of the virus, that is a theory which has never obviously been seen. All we know is there's no virus to see in the beginning. And then once the tissue is dying, we see lots of these little particles of various sizes and shapes. And even if you look at that slide that person showed, I'm sure if you looked at other parts of the slide, you would see many other different types of particles. And so why is it that we're saying that this particular particle is the cause of the disease and not some other particle that you would also see there? Because there is not a uniform morphology of the particles. Now, the other thing we have to realize is that one of the problems we're getting into with modern biology, and I mean modern biology over the last 100 and some years, is that we only really study dead tissue or dead cells. And in particular, with the era of virology, we study them and we see them, like in that picture we saw, only under an electron microscope, because they're too small to see under a light microscope. So even that picture, that is not a virus, that is a heavy metal stain of something, which we then call a virus. If you realize how an electron microscope picture is made, which is basically the tissue is dead, which changes it, and then the tissue is frozen, usually at 100 and some degrees below 0 centigrade, and then it's stained with heavy metal stains, and it shot an electron beam at it, which immediately evaporates the water. The question is, how reflective of what was actually present in the living system, in the living being, is that picture? I mean, if I said to you, I want to find out what your hand is made of and what it does, and the way I'm going to do it is cut your hand off your body and put it in an enzyme bath and freeze it at 150 degrees below zero, and then put it in a blender, and then shoot an electron beam at it, and then put it in a heavy metal stain. And I see basically a picture of it under electron microscope. I don't think anybody would say that that is a reflective of what your hand looks like, or that your hand could stroke a baby and make it feel better. So we have this illusion that somehow these pictures that we see under an electron microscope are reflective of reality. And so when you get to something like ribosomes, which are an integral part of this whole theory of DNA being made into protein, being translated into, DNA being made into RNA, being translated into protein. The whole thing happens on the ribosomes. And the only way the ribosomes have been proven to exist really is to be seeing them in electron microscopes. And they always look like perfect circles, which means they must have been perfect spheres in real life, which means that you've somehow put this in a blender, and there's no way that you would get only perfect spheres or perfect circles if you blended some sphere up. And the person who really is the master at dissecting this, who I've talked about, is somebody named Harold Hillman, who is a British biologist. And for anybody who wants to understand what is real and what isn't real in cell biology, I think you have to read Hillman, because he was the only one that I know of who really went through step by step and tried to figure out what was artifact and what isn't. And at the end of the day, he said the only thing you can actually see in a living tissue, in a living cell, is a very thin cell membrane that looks like it's fat soluble. You see organized, or sometimes called coherent or structured, water. You see mitochondria, which look like little threads in the light microscope, and you see a nucleus. And everything else, I would say, including endoplasmic reticulum and ribosomes and Golgi apparatus, is suspect because of the way that they were found. And I certainly wouldn't expect anybody to believe me, but after reading 1,500 pages at least twice of Hillman, I think if anybody wants to engage in this debate, your first step is to read Hillman thoroughly, see his evidence, and then you'll have something that's more appropriate and scientific to say about what is real and what isn't.
Okay, thank you. And it's also my understanding that no one has ever seen a live virus under a microscope.
Well, no one has ever seen what they see is no particles in a normal person, and then lots of these particles under an electron microscope of various morphology, sizes, constituents, little spiky things under an electron microscope after they're sick. All the steps in between, the binding with the receptor site, the injection into the cytoplasm, somehow the transfer of that material into the nucleus, the emergence out of the nucleus into the cytoplasm, the budding out of the cell to emerge with thousands and millions of these particles, that has not been seen like in a movie. Those are basically the viral theory to account for all those steps in between.
Thank you. Thank you. Okay, so we have maybe time for a few more questions. Renee, would you unmute yourself, please? Okay. You had yourself unmuted. While we're doing that, let me just ask this question. There's been a lot of talk about the damage to mRNA delivery systems, what it can do to cells. Okay, and can you elaborate on how that damage can occur when delivered by mRNA vaccines?
Tom, do you know what she's getting at there? I'm not sure I understand this question.
No, I didn't quite follow that.
Yeah, so just trying to read. I believe it's in regard to cancer cell growth and hormone endocrine disruptors. Okay, maybe that's a little disjointed the way that it was written, so I apologize for that. So I believe, they're talking about how the vaccine is going to change people's RNA or their DNA. And a lot of that was addressed in the presentation.
Well, the truth is we really don't know if it will change the DNA or RNA at all. We have talked about the possibility that if the RNA can be intact, as Tom pointed out, because, the reason they have to transport it at such a low temperature is because, as soon as RNA gets to room temperature, it basically disintegrates. So if it can actually go, into a cell, it's possible it could be integrated into the nucleus some way through reverse transcription. Certainly the DNA with the adenovirus, the Johnson & Johnson, that is intended, at least to the description that scientists have about that type of technology, that it's supposed to go into the nucleus and be integrated into the host DNA. Whether that actually happens, we really don't know. There's no studies that have been published that show what happens to people after they've been vaccinated.
I see. Okay. Thank you. All right, Renee, you're unmuted now. What's your question? Okay. Hello? Okay, thank you. We're going to try it with another guest. Thank you very much. All right, so recently Rand Paul was grilling Fauci on his involvement in gain-of-function research in Wuhan lab. If viruses do not exist, as we have been told, what is this gain-of-function all about? Is it just more fiction for public consumption and building the narrative?
Well, it certainly is a situation that could generate a lot of fear, because if you think about, these scientists under government contracts developing weapons, right, that if they unleash those, they would be devastating, right? That's the kind of the fear that's generated. But since there's no, natural model for, some kind of airborne germ or anything like this, to me, it seems most likely that this is just vaccine research that they're doing, and that the vaccine actually is the weapon.
I see. So maybe, let's just kind of wrap up talking about how do we talk to our families? Our families are going out there and getting vaccinations, and they think it has to be done. A lot of people are asking, how do we what do we do when our brothers and sisters and mothers are going to get vaccines? How do we start talking to them, and what are the things that we can really impart to them to get them to consider, doing something else?
Tom, do you want to speak to this? I mean, I'd be happy to.
Yeah, I mean, what we've learned through this whole thing, and I think we've learned it in this culture for years, is that the way people solve problems is they basically shout at each other. And the way they communicate is they shout and they espouse their own ideology. And they don't do the experiment of, is it the Star Spangled Banner or is it the guns that are killing the prisoners? So the way out of that is tough. But I think it's to ask people how they came to this conclusion. Because I think if you just say your theory, your ideas, or your understanding, if somebody isn't able or willing to hear that, it will just fall on deaf ears. So it may be better to try to have people explain how they came step by step, which is one of the reasons why I've now started with this question: how do we know there's a virus and that the virus causes disease? because it seems to me that if you can't answer that question, it's really hard to imagine why you would want to get an injection for something which you have no idea whether it even exists or is a problem for you. Now, you may say, well, I believe certain people, and that's fine, but you may ask them why you believe those people. And ultimately, this is about freedom. And we can't convince anybody to do anything, nor should we. You can only say what's true for you and then let the chips fall where they may, and realize that we're all at different places for whatever reason in our destiny. And we have to somehow honor and respect that and hopefully move on.
Beautifully said. Thank you. All right. So let's see. Benedict, can you unmute yourself? We're ready for your question.
Let's see, does that work?
Yes, hello.
All right, all right. Hey, first up, guys, thank you. Big thanks to you guys plunging in here and taking what it takes to really delve and expose this bullshit that's been going on for a couple of 100 years, right? And, it's like everybody that's been licensed has an issue now, right? And you have broken out of that, and thank you guys for really pioneering this. I knew this was all bullshit back in January 2020. So my question here is, as a health coach, I really expose what I believe to be true about germ theory myths right at the beginning. The question here is, having worked with sound and electrical healing for over 20 years, right? Developing my own sound healing devices and then electrical step devices. My question is, as our doctors who are looking at all of this whole envelope of issues that we're faced with, not only the medical, but the legal and everything else, why is it that doctors are not really emphasizing the importance of fasting and really boosting the immune system that way?
I mean, when you think about how, Andy and I are both, or were anyways, licensed medical doctors, and we were both trained to be medical doctors. And, science and medicine is an interesting philosophical dilemma, because when you talk about sound healing and energy healing and waves and things like that, like I say, at the end of the day, if I think about what I know to be real, as I said, it's a little compartments with lipid envelopes, I think, with organized water, a nucleus, and a mitochondria. And the whole thing is organized through the water, which is the main player in this, is organized through being a receiver for energies coming from the outside. Essentially, our tissues are like radios. And so the sound doesn't come from inside the radio. It comes from, the radio antenna beaming the sound. But obviously you have to have an intact radio in order to receive that wave, that sound wave, and transform it into something we can hear. That role is played by water in our tissues. So that's the connection. But here's the point to answer your question. If you, as in all your training as a medical doctor, say anything about anything else being real but purely physical substance, then you are denigrated and laughed at, period. Now, I've often thought about this. So the theory of our science, the theory of our medicine is only physical substance exists. And I've often thought about this. So if you take a heart surgeon, again, let me just say that again. He thinks, he's been taught that only physical substance exists. In fact, we think that the mind, which is asking the question, how am I thinking about this? Or how do cells create these words and thoughts that I'm saying now? Somehow that comes out of the brain cells. We have no idea how that happens, but we all believe it. So you ask that you go to this heart surgeon. So you only believe in physical stuff, right? Yes. So do you love your wife? Yes. How much? With all my heart. Hopefully you'll say that. Now, I can tell you, if you dissect somebody's heart cell by cell, I can make a very accurate prediction that you will not find anything called love in that person's heart. And so my point is, we actually all live our lives as if that theory wasn't true, including doctors. They know that's not true because it's obvious that essentially human beings are quality sensing devices. You know, we don't marry this person instead of that person because they have 6 more hydrogens and 2 fewer sulfur atoms in their body. But yet that's all we say exists. And I mean that. That's all we say exists. Never mind that sometimes they're particles and sometimes they're waves, and we don't even know how that somehow forms into physical stuff. And so it's like we're working on a theory that nobody has proven, and none of us actually believe, but that becomes the entire philosophical basis of science and medicine. And even it gets into addressing this person's question, because I've thought about this. How is something transmitted through a cell culture? So you start with all kind of toxins, and then potentially you dilute them. But we know from homeopathic research that if you take something and you dilute it over and over again, it still retains some sort of energetic imprint in the final solution. But the problem is, if you introduce that into the conversation and say, these energetic imprints, which is probably sort of the whole basis of science and medicine in the first place, that's real. They say, no, you cannot, that cannot be even talked about. You cannot even investigate that because that's not part of what we accept as science. My point here is, if you accept as science that only physical matter exists, only that which can be measured exists, you are sunk, because that's not how human beings operate. That's not what a living system is. Essentially, that's saying we have no theory that explains the difference between a living thing and a dead thing, which is what Goethe said. We went wrong when we decided to understand the frog, and the first thing we did was kill the frog. And at that point, there's nothing to understand about the frog.
Thank you. Thank you. It's very good. I like your analogy, with the heart, and you'll never find it, just like you'll never find consciousness if you dissect the brain. That's
never. It's not there. The substance is made from consciousness, not the other way around.
Thank you. Okay, this is somewhat of an esoteric question. Now, doctors, we are at a certain point where we have, we've gone a little over. How is everybody right now to take a few more questions? And, what's your thoughts on, doing a little more? Or should we do one more?
I think we should. I think that's probably enough, because you start getting into wonky answers after a while.
Yes.
And so if there's interest, maybe we should do it again. But after a while, it starts getting fuzzy around the edges.
Yes. Okay. Well, I think we're there now.
Yeah.
Okay. Great. Well, this was just fantastic. I think everybody got a lot of answers to their questions, and the presentations were outstanding. Okay.
Well, thank you so much, Leah. We really enjoy working together, and every time I hear Tom, I'm just kind of taken back listening to the wisdom and the way that he puts things together. And I know everyone else is having that kind of moment as well. I want to remind everyone that we have the bonus materials are still available in the Files tab. and just see, Leah, do we need to give any instructions or anything out before we wrap things up?
Yeah, there's a few things coming up. So you will get the downloadable material via email. If you didn't get a chance to download it here in the player, or if you're watching this on replay, that will be made available. Okay. So we have some upcoming events. We have the End of Germ Theory with Jamie Ike and Dr. Andrew Kaufman. So you can register for this live event by going to www.iconic.com and clicking on live events. Okay. So hold on a second. Let me just mute everybody because I'm getting feedback. Okay. We also have the True Healing webinar Q&A with Dr. Andrew Kaufman. Go to andrewkaufman.com, select live webinars. And this is a webinar series where Dr. Andy will discuss using nature to heal your own body and becoming your own health authority. Please visit drtomcowan.com and join Dr. Cowan for Conversations with Dr. Cowan and Friends, where Dr. Cowan and his guests discuss forming a common sense approach to medicine and biology grounded in deep inquiry and observations of nature. Dr. Tom will be presenting at Experience Polyface on June 18th through 19th. Go to polyfaceyum.com for more details. Both doctors will be presenting at the 3rd Annual Advanced Medical Conference in Sioux Falls, South Dakota. Dr. Andy will be live, and Dr. Cowan will be presenting remotely, and it'll be a joint presentation back-to-back, May 29th and 30th. And while Dr. Andy is in South Dakota, he will also be presenting at the Red Pill Expo in Rapid City. We want to thank you to the planning teams from both camps. You guys were amazing. Thank you to the technical team. You pulled us through. Some people will be catching this on replay. You know, we were under attack, and we had people working on this. We got everything up and running. So thank you all for your kind patience and your support. The proceeds from this webinar will go to continuing the research of both doctors. So we thank you very much for that. So thank you for everyone who put in this amazing effort to make this happen. Thank you for everyone that joined us today. And thank you, Dr. Andy and Dr. Cowan, for your efforts in bringing truth to humanity and fighting for our freedom. Okay. And we're going to have to unmute you both to talk.
Sorry, yes. Thank you, Leah, for those closing remarks, and everyone for all your help. And of course, most of all, to all of the participants and guests. I hope that we brought you, a program with good value today. And please do take advantage of, looking at the replay material that we will send forward. And, we will look forward to seeing you again in the future. All right. Signing off. COVID-19 Myths 2021.
Peace.